Very good evening, my dear friends. So today's resource person, our CPGET 2022, Dr. K.RANI Madam Garu, Assistant Professor, Department of Botany, Government Degree College, Siddipet Autonomous. Madam is going to explain today's one of the important topic in photosynthesis of plant physiology, that is CAM pathway. Madam, please start your session, share your screen.
Oh, okay, sir. Thank you, sir. Thank you for giving me this opportunity. Sir, host disabled participant screen sharing. Give me the host, sir.
Already given, madam. Once you
got it, sir. Okay, sir. Are you able to see the slide, sir?
better if possible, please click on slide, enlarge it.
Okay. Okay, sir.
లేదు మేడం మనం
Very
లేదు మేడం మీరు ఒకసారి క్లిక్ చేయండి, డబుల్ క్లిక్ చేయండి.
చేశాను, సార్.
వస్తలేదు.
Okay, sir. This is Dr. Rani from GDC Siddipet. Last two classes, we discussed that in the photosynthesis, as part of photosynthesis, we are discuss about that the dark phase. So photosynthesis is completed in the two stages, as I told you. Almost the light phase is completed, and in the dark phase, last two classes, C3 and C4 also completed. Today we are going to discuss about that the CAM pathway. CAM stands for crustacean acid metabolism. Crustacean acid metabolism. So almost it is an adaptation for increased efficiency in the use of water, and so it typically found in plants. They are growing in the arid conditions. Almost the leaves in the plants of some crustacean families like Kalancho or Sedum. So they all are the best example of the crustacean family members, and some certain members of the cactus. Cactus also, they are belongs to the CAM plants, CAM plants. So cactus, orchid, and some pineapple. These families are characterized with succulent habit, and they will show a diurnal pattern of the organic acid formation. So almost it is an alternative pathway of the CO2 fixation, and CAM plants are the adaptation for increased the efficiency in the use of water. So they are almost, these plants are typical plants. They are founding in the arid conditions only. Almost all CAM plants, they are the succulents. All the CAM plants are the succulents, but all the succulents are not CAM plants. So this is very important in entrance point of view. All CAM plants are the succulents, but all succulents are the not CAM plants. So I will give some examples. Almost cactus, kalanche, sedum, and pineapple. So some orchids. These are families are the characterized the with succulent habit. So almost these plants, they will show the diurnal pattern of organic acid in their life. These plants, they are an adaptation to xerophytic life. So almost these plants, they will show the xeroc conditions or xerophytic characteristics. And these plants, their life close their stomata during daytime, and stomata will open during the night time. So this is the main characteristics of the CAM plants. So almost these CAM plants in their life, so the stomata is open during the night time only. Stomata will close during the daytime. So the stomata is open during night time. They are called as the photoactive stomata. So almost all the plants will show that photoactive stomata. That means during daytime, the stomata will open. That is the photoactive stomata. But these CAM plants, so the stomata will open during night time only. So this stomata is known as a photoactive stomata. So in these plants, the carbon dioxide fixation takes place in night time. Hence, this method is called the dark carbon assimilation. So almost in C4 plants, so that is the cycle will completed in two plants, chloroplast dimorphism. As I told you that mesophyll cell and bundle sheath cell. So as per same, in the CAM plants, the CO2 fixation takes place in night time only. So this method is called dark carbon assimilation pathway. Okay, so this pathway has been identified in 20 angiospermic families. As I told you, some examples like Crussulaceae, Cactaceae, Agaveaceae, Orchidaceae, Portulaceae, and Euphorbiaceae. So these all are the angiospermic plants. So almost this CAM pathway has been identified in 20 angiospermic families. So almost this cycle will completed in two during daytime and night time. So I will show the cycle. This is the cycle, Nana. So just wait. So comparison of the C3, C4, CAM. Okay. So the CAM pathway will completed in the during daytime and during night time. Okay. So these plants, almost to the CAM plant leaves, they will show a decrease in total organic acid content with an increased pH of the cells of during the daytime. So while almost this diurnal pattern is accompanied by a closing and opening. It's based on the closing and opening of the stomata. So almost so in this way, almost the CAM plants will discuss that photoactive and scotoactive stomata. So in this, the carbon dioxide also fixation in the night time only, during night time. So in that, almost it occurs mostly in succulent plants, as I told you. So they are growing arid conditions. So this cycle was first discovered in the plants belonging to family Crussulaceae. The CAM cycle is completed in two parts, Nana. So in that first one is the acidification part and second one is the de-acidification part. Just I'll show the screen. CAM cycle that is completed in two parts. So acidification and de-acidification. Acidification takes place in dark time, or acidification takes place in the dark, while de-acidification occurs during the daytime. So first we'll discuss in the during night time only, or some the first step of the CAM pathway. So during the night time, a plant employing the CAM has its stomata. It will allowing carbon dioxide to enter and it will be fixed as organic acids by a phosphoenol pyruvic reaction similar to the C4 pathway. So yesterday we'll discuss that some first step of the C4 plants. They are the almost in the C4 plants, the first stable or first CO2 acceptor is the PEP, phosphoenol pyruvate. So it will combines with the carbon dioxide and convert as a C4 carbonous compound. So as per same, during the daytime in CAM pathway also, it has the stomata will allowing the carbon dioxide during night time. So the CO2 entering into the basophil tissue of the leaf. So the organic acids observe the phosphoenol pyruvic reaction. So as per same C4 pathway. In the first step, the resulting organic acids are stored in the vacuoles. Just the cycle is missing, I think. So this is the CAM pathway. You can see here the CO2 is entered into the night time. Why night time? Almost the leaf stomata will open during night time only. So this is the main characteristics of the CAM. So the stomata will open during night time. So for that purpose, the CO2 will absorbing through stomata into the basophil cell. So in the leaf cell, it is the content of the phosphoenol pyruvic acid absorb the CO2 to modify C4 compound, that is oxaloacetic acid or malic acid. So that is the C4 compound. And in this almost they show the maximum gases exchange at night because of they are showing the nocturnal opening of the stomata. So in this, in the acidification pathway, the stored carbohydrates are converted into phosphoenol pyruvic acid through the glycolysis. So in this, the CO2 diffuses freely into the leaf through the open stomata in night time. So for that purpose, the phosphoenol pyruvic acid combines with carbon dioxide and water molecule also are adding to these substances to convert as a oxaloacetic acid and H3PO4. And in the second step of the CAM pathway, the oxaloacetic acid or the C4 compound is now reduced to malic acid in presence of the enzyme malic dehydrogenase. Please, you can observe the CAM cycle only. After that, we will discuss the comparison of the C4 CAM cycles. Okay. So almost these all reactions occur during night time only. Night time is acidification reactions. So in the first step, the phosphoenol pyruvic acid combines with atmospheric carbon dioxide and water molecule also combine to this compound to convert as a oxaloacetic acid and H3PO4. So as per same, in the second step of the CAM pathway, the oxaloacetic acid is now reduced to malic acid in presence of the enzyme malic dehydrogenase enzyme. So this reaction is facilitated in presence of reduced NADP plus. So that is almost it will convert as a malic acid plus NADP. NADP plus H plus is reduced to NADP plus. So in this, almost the malic acid that's produced in dark as a result of acidification that is stored in the vacuole. So the oxaloacetic acid may also be interconverted into aspartic acid. So almost these two reactions occur during night time. And the pyruvic acid, so it will formed in the reaction that is either oxidized to carbon dioxide through the other C3 cycle. So it is reconverted to, or in other way, so it is also phosphoglyceric acid to synthesize sugar through the C3 cycle. So in this cycle, almost the CO2 liberated by deacidification of malic acid that is accepted by ribulose biphosphate to fix it into the carbohydrate through the C3 cycle. So however, almost the fate of the pyruvic acid is still not clearly known in the C4 pathway. That is the CAM, just we will discuss in the text part. During night time, a plant that is employing the CAM as its stomata and it allowing the carbon dioxide to enter and it will be fixed as organic acid by phosphoenol pyruvic reaction similar to the C4 pathway. So the resulting of organic acids, they are stored in the vacuole they are entering into the almost the CO2 is eliminated from that. It is participate in the Kelvin cycle. So it cannot operate without ATP and NADPH. So for that purpose, it is a light dependent reaction that do not take place at the night time. And next, so during daytime reactions, almost this cycle is completed during night and during daytime, as I told you that. So after night time, after acidification reactions, then the deacidification also occur during the daytime. So in the deacidification means during the daytime, the stomata will close to conserve the water and carbon dioxide also storing organic acids. They are released from the vacuoles of the mesophyll cells. So in this, an enzyme in the stroma of chloroplast, they are releases the carbon dioxide. So it enters into the Kelvin cycle. That photosynthesis may takes place in the daytime. So this is almost the C4 pathway bears the resemblance to the CAM. That means what is the comparison? I think the C4 or the CAM is look like same, just to there look like a resemblance of the C4 pathway. Both pathways act to concentrate the carbon dioxide around the ribulose, bisphosphate, carboxylase and oxygenase. So thereby increasing its efficiency. So that is the main. So the CAM, crussylation acid metabolism, they will concentrate it temporarily. It will providing the carbon dioxide during the day and it not at night. So when respiration is the dominant reactions in this way. And in the C4 plants, in contrast, they will concentrate the CO2 spatially with a Rubisco reaction center in the bundle sheath cell. So yesterday we'll discuss that mesophyll cell and bundle sheath cell. So they both will, they are being inundated with the carbon dioxide due to the inactivity required by the CAM mechanism. The C4 carbon fixation has a greater efficiency in terms of the phosphoglyceric acid synthesis. So what is the significance? What is the importance or the summary of the CAM pathway? Almost this the plants, the CAM plants and C4 plants, they will resemble to each other, as I told you that. So the CAM plants, according to this plant, there are 2 carboxylations will occur. So in this, the CAM cycle, almost it gives you a simple reversible acidification and deacidification. According to the cycle or CAM pathway, the carboxylation as in the Kelvin cycle and phosphoenol pyruvic carboxylase. So while this both hypothesis lacks the decisively experiments, but it has some special or some appealing aspects differ from the C4 and C3 plants. So almost the main 2 points is differ from the 2 cycles or the CO2 fixation. It gives you a simply reversible acidification. CAM pathway gives you a simply reversible acidification and deacidification. So which is definitely characteristics of the physiological process. And almost in the second point of the to differentiate, the phosphoenol pyruvic, it seems to be likely an intermediate. So because this is the only known gateway in going from organic acids to carbohydrates. So this is 2 main points to differ from the C4 and C3. And CAM plants and C4 plants both will resemble to each other in showing the CO2 assimilation. In both plants, they will incorporate the reductive pentose phosphate and C4 dicarboxylic acid pathway. So, but in C4 plants, the 2 cycles are found separated, one in mesophyll cell, while other in the bundle sheath cell. So, contrarily, in plants, they will both pathways are found in the mesophyll cells, but the timings are differ. That means almost during daytime and during night time, the both acidification and deacidification will takes place in the CAM pathway only. So that means the CAM pathway is similar to C4 pathway in that carbon dioxide, that is trapped by highly efficient phosphoenol pyruvic carboxylase enzyme. So, however, instead of having two kinds of photosynthetic cells, the CAM plants has only one kind of photosynthetic cell in which CO2 is fixed during night and used to make glucose during the daytime. So this is the main summary of that CAM pathway. Okay, so the CAM plants live in very dry condition, and unlike other plants open their stomata to fix that carbon dioxide only at night time only, and so the oxaloacetic acid is converted to malate, which is stored in the vacuole. So during daytime, when the stomata are closed and CO2 is removed, the stored malate and enters the Kelvin cycle. So this is main. And during almost these plants, they are drought resistant and they can possess a xerophytic adaptations like thick fleshy leaves. And another adaptation of the CAM plants, so they are able to fix carbon dioxide in dark. They can survive for longer periods in light without CO2 uptake. So this is also main significance of the CAM plants. And in these plants, so the stomata of leaves, they will remain closed during the day and open at night time. So this is also an adaptation to conserve the water. Since the succulents exhibiting the CAM, they are found in the dry habitats also. So almost there are some C4 CAM intermediate species. We'll discuss that. And what is the CO2 compensation point? So this is in examination point of view, CO2 compensation point. So the compensation of CO2 at which the rate of respiration, that means the release of carbon dioxide, and rate of photosynthesis or the consuming or utilization of CO2 are equal, that is called CO2 compensation point. So this is examination point of view, MSc. So the concentration of carbon dioxide at which the rate of respiration and rate of photosynthesis both are equal. So that is called CO2 compensation point. And at CO2 compensation point, the net gain of photosynthesis is 0. So this is also very entrance point of view important. Almost CO2 compensation point is the net gain of photosynthesis is 0. Okay. So the CO2 compensation point is less in C4 plants, just like 1 to 5 ppm of carbon dioxide, and that is more in C3 plants. So almost 40 to 60 ppm. Okay. Hence the photosynthetic rate is more in the C4 plants. How the photosynthetic rate is more in C4 plants means the CO2 compensation point is less in the C4 plant. So that's why, as usual, it is against that. So the photosynthetic rate is also more in the C4 plants. So this is almost what is the difference between the characteristics of C3, C4, CAM plants. Okay, close. Okay, the difference between the C3, C4 and CAM plants. This is only, I'll take the main important characteristics, the leaf anatomy. First one is leaf anatomy. So leaf anatomy, the cranch anatomy will observe in only C4 plants. So why it is cranch anatomy? Almost we'll discuss yesterday. The leaf mesophyll tissue have the chloroplast, dimorphism chloroplast will observe in that. One is the mesophyll cell chloroplast and the second one is the bundle sheath cell chloroplast. So the size also large in the bundle sheath cell, in bundle sheath cell. So that's look like a wreath-like structure. So flower-like structure. Kranz means in Germany. So that is the flower vase or wreath. So that's why in the monocotyledon leaf anatomy, we'll observe that the vascular bundles are arranged in the mesophyll cell, look like a flower vase type. That is called cranch anatomy. So the cranch anatomy will observe in the C4 plant. C3 and CAM is they will not having that cranch anatomy. And second one, catalyzing enzymes. So first, which enzyme is a key enzyme, or which enzyme is very important in the cycle? So in the C3 plants, RUBP case, ribulose bisphosphate case, and almost C4 and CIM both will same. They are in the mesophyll cell have the phosphoenol pyruvicase. Okay. So C3 is RUBP or Rubisco. Just we can remember that Rubisco is important in the C3 plants. It is very key enzyme. That is protein enzyme, RUBP, and remaining C4 CAM will have that to PEP case. And in the primary acceptor, almost the CO2 acceptor of the C3 is RUBP enzyme. So, and CO2 acceptor of the C4 plants, both will C4 and CIM, that is the phosphoenol pyruvate. So that is the first primary acceptor of the C4 and CIM. And in the first stable compound of the C3 plants is 3-carbonous compound, that is phosphoglyceric acid. And in the C4 plants, same oxaloacetic acid, malic acid, aspartic acid. So these all are the 4-carbonous substances. As per same and CAM plants also have that malic acid or oxaloacetic acid. And in the another important characteristics of the point is photorespiration. So the photorespiration is presenting in only C3 plant. So C4 and CIM in the two absent. So they do not have that photorespiration. And the CO2 compensation point, that is the almost all the same, less than or greater than 50 almost. And the carbon assimilation rate. So the carbon assimilation rate is less in the C3 plants, more in the C4 plants, and moderate or less in the CAM plants. So the optimum temperature for photosynthesis, almost C3 is 15 to 20, and these all are the arid conditions. So that 30 to 35 degrees centigrade of the C4 plants, but in the CAM plants have the almost, they have the less 10 to 25 degree centigrade.